Now this is a continuation from the three-part series I've done. The first one being the overview of photosynthesis, the second one being the light-dependent reaction, and this one being the light-independent reaction, AQA Biology. Key terms. The light compensation point is when carbon dioxide is absorbed by photosynthesis is exactly balanced by the oxygen absorbed and the CO2 by cellular respiration. And then the rate of photosynthesis is the same rate as that of respiration. Products and site. The products of the light-independent reaction are ATP and reduced ADP and ADP. Sorry, I meant the light-dependent reaction. Sorry about that. And the site of the light-independent reaction is the stroma. The Calvin cycle. Now, like I said before, the light-dependent reaction takes place during the electron transfer link. Now, this is a little bit after that. So you have the products of the light-dependent reaction, which is the ATP and the reduced ATP. And it goes into 3 stages. So the stage 1 is carbon dioxide fusing with RuBP. And ATP and reduced NADP reduces, no, sorry, one second. Carbon dioxide forms with RuBP. And this forms a 6-carbon compound. This 6-carbon compound forms GP, which is a 3-carbon compound, and it forms 2 of these. Then ATP and reduced NADP reduces GP to TP, and then RuBP is regenerated. So I'm going to show this as a little cycle as you would get it in the exam. So you have RuBP. I hate that B. Let's do that again. B. I would have left the diagram, but there wasn't a good one that I could find, so gonna have to make do with this. So you have carbon dioxide coming in. Now you might be wondering, where did the carbon dioxide come from? Well, remember you have the open spaces in the leaf in which carbon dioxide can enter and water leave. Then you have GP, which is your 3-carbon compound. And you have 2 of these that are made. Then this is reduced to form TP. Which is also a 3-carbon compound. And this is done using ATP. ATP. And this turns into ADP plus PI. As well as this, you have the same thing occurring for reduced NADP. So I'm going to write it as NADPH Actually, let me not write it here because you can't really read that. I'm going to do it underneath. So NADPH I wouldn't recommend you writing H, but I'm not going to write reduced because this is the struggle. Forming NADP. Remember in your exam, write reduced NADP because you can lose marks if you write the formula wrong. Whereas if you write reduced NADP, you'll get the marks. Then TP reforms RUBP. And keep in mind, as this is actually 6 and this is 5, it means that 1 goes into useful products. So let's call the useful products X, because I'm not going to write out useful products, but I'm going to write out useful. All right, so we're going to go through this again. So you have your RUBP, a 5-carbon molecule. It binds to carbon dioxide, forming GP, which is a 3-carbon molecule, and 2 of them are formed. Then using ATP and reduced NADP, they form ADP plus PI and NADP, and this forms TP. This reduces GP from TP, which is another 3-carbon molecule. Now 5 of these carbons go to reform RUBP, and 1 of them are useful products. And as you're trying to make glucose, you'd have to do 6 turns of this cycle in order to make 1 glucose molecule, because the useful products make up glucose. And if you can only get one useful product in each turn, you need 6 carbons in order to form glucose. Because remember, the formula for glucose is C6H12O6. Hopefully that made sense. If it doesn't, then please rewind the video and watch it again until it makes sense to you. For you. Use of equipment measuring limiting factors. Well, limiting factors can be measured in the form of light intensity, temperature, and carbon dioxide. Now you might be wondering, why are these limiting factors? Well, if you have a limiting amount of light, then that means photosynthesis can't occur. If there's a low temperature, enzymes denature, and carrier proteins like NADP, NAD, and the electron transport link can't occur because they're made up of what's the thing them called again? Proteins. And if proteins are heated to a higher extent or not enough, then they start to denature. And if there's not enough carbon dioxide, the carbon dioxide can't bind with RUBP. And if they can't bind with RUBP, then the reaction cannot occur. Now, in your exam, just a little tip is that RUBP is also known as Rubisco. So if you hear about Rubisco in your exam, don't get confused. Rubisco is RUBP. So that's all there is for photosynthesis, and the next video will be about respiration, more specifically glycolysis. So if you like this video, then please like and subscribe, and I'll see you on the next one.